Assalamu alaikum. Today we will study about the nucleic acid called deoxyribonucleic acid or DNA. Its definition is it is a nitrogen containing compound, a macromolecule that carries genetic information and it is present in the nucleus of all the cells. Its function is cell division. We know that the new cells are made from the pre-existing cells when they divide. Second function is cell differentiation. When the new cells are made, they are differentiated into different types of cells like neurons, which are the cells in our nervous system, muscle cells, red blood cells, etc. The structure of DNA is that basic unit in the structure of DNA is called the nucleotide. Nucleotide contains these three things: a sugar called deoxyribose sugar, which is a 5-carbon sugar, a phosphate group, and a nitrogen containing base. There are 4 kinds of nitrogenous bases: adenine, guanine, cytosine, and thymine. And these 4 types of nitrogenous bases, when they combine with the phosphate and the sugar, they form the 4 types of nucleotide. So we have adenine nucleotide, guanine nucleotide, cytosine nucleotide, and thymine nucleotide. These nucleotides are joined together and they make the long chain of nucleotides, which are called the polynucleotides. And the 2 strands of polynucleotides are twisted around each other, called the double helix structure of DNA. Thank you. Students, the diagram shows the building block of nucleotide. The nucleotide is made up of the 4 types of nitrogenous bases. This one is the adenine base, this is cytosine base, this is guanine, and this is thymine base. There is a sugar, deoxyribose sugar, the 5-carbon sugar, and a phosphate group. Now, these 4 types of bases, when they combine with the base, the deoxyribose sugar and the phosphate group, so 4 types of nucleotides will be formed by the 4 types of bases. This is adenine nucleotide, thymine nucleotide, guanine nucleotide, and cytosine nucleotide. And these nucleotides, when they are joined together, they form the long chain of nucleotides, which is called a polynucleotide. Here we see the 2 polynucleotide strands, and there is a rule for matching of the base pair of these 2 polynucleotide strands. According to this rule, the guanine of one polynucleotide strand, it will match with the cytosine of another polynucleotide strand, and the adenine of one polynucleotide strand will match with the thymine of another polynucleotide strand. And here in this diagram, we see the double helix structure of DNA, in which the 2 polynucleotide strands, they are twisted together like a helical spring. And this structure, the double helix structure, this was discovered by Watson and Crick, the 2 scientists. And here, this is the untwisted form of the DNA model or DNA structure. There are 2 polynucleotide strands, and this blue strip shows the deoxyribose sugar and the phosphate, and inside these 4 colored nitrogenous bases. And here with this polynucleotide strand, this is also the deoxyribose sugar and the phosphate of this polynucleotide strand, and these are its nitrogenous bases, and these bases, they are combined with the same rule, that is the guanine matches with the cytosine and the adenine matches with the thiamine. You can learn this rule that guanine matches with the cytosine GC, the government college, which has turned into Government College University, Lahore. So this is the double helix structure of DNA. Now you can interpret this diagram in a more better way. Thank you. Students, we have another type of nucleic acid called ribonucleic acid. Ribonucleic acid, or RNA, is made by DNA to transmit the genetic information from nucleus to cytoplasm. You know that cytoplasm is the part of the cell where the chemical reactions are taking place. How RNA is, now we will study the difference of RNA from DNA. RNA contains the base uracil instead of the base thiamine. We know that there are 4 types of nitrogenous bases in these nucleic acids, and instead of thiamine, the RNA contains the nitrogenous base uracil. And in RNA, there is a single strand of polynucleotide, and there is ribose sugar instead of deoxyribose sugar. There is a difference in the ribose sugar and deoxyribose. In deoxyribose sugar, there is 1 oxygen atom less than the ribose sugar. Ribose sugar will have 1 more oxygen atom. Both are having 5 carbon atoms. Both types of sugars are having 5 carbon atoms, and these are 5 carbon sugar. Now we can write the differences between the DNA and RNA in this table form. DNA contains 2 polynucleotide strands. RNA contains 1 polynucleotide strand. DNA contains deoxyribose sugar. RNA contains ribose sugar. DNA contains the base thiamine. RNA contains the base uracil. DNA remains inside the nucleus, whereas the RNA is made by the DNA and it goes out of the nucleus in the cytoplasm with the information given by the DNA to make the protein as given in the information of the DNA. These proteins, they are making our characteristics.